Welcome to this ASN Kidney 360 podcast. I am Karl A, Professor of Medicine, Division of Nephrology and Hypertension, Mayo Clinic. I am joined by my co-author, Dr. Vesna Garovic, Professor of Medicine, Chair, the Division of Nephrology and Hypertension, Mayo Clinic. Today we will discuss our article that is entitled Expression of ACE2 in the Intact and Acutely Injured Kidney, published in Kidney 360 this July. The COVID-19 pandemic affected everyone, and in a sense, COVID-19 led us to undertake this study. In 2020, it was discovered that the causative SARS-CoV-2 virus entered cells via a receptor identified as angiotensin-converting enzyme 2, ACE2. Following entry, ACE2 expression decreased. ACE2 was discovered 20 years previously in 2000 as a counter-regulator of the renin-angiotensin-aldosterone system. ACE2 produces angiotensin 1,7 from angiotensin 2, and angiotensin 1,7 exerts vasodilatory, anti-inflammatory, and anti-fibrotic effects. These effects oppose those of angiotensin 2 and aldosterone. Prior literature indicated that ACE2 is cytoprotective in tissues, and the field of nephroprotection has long been of interest to us. Furthermore, SARS-CoV-2 infects the kidney, and in COVID-19, AKI is a recognized complication which worsens morbidity and mortality. We thus decided to study the significance of this enzyme in AKI, and the current paper assesses ACE2 expression.
Thank you, Dr. Nath. Can you please briefly describe your experimental protocol?
Thank you, Dr. Garovic, for joining me on this podcast. As regards your first question, we examined ischemia reperfusion AKI, which is characterized by cell necrosis, inflammation, and reduced GFR. Lipopolysaccharide LPS-induced AKI, which models sepsis, and urinary tract obstruction, in which GFR ceases with relatively little in the way of necrosis and inflammation. We determined whether the heme oxygenase system was involved in determining ACE2 expression after LPS. We assessed the expression of the MAS receptor, the receptor that mediates the effects of angiotensin 1,7, the putatively beneficial product of ACE2. As male sex and age predispose to AKI, we assess the effect of sex and age on renal ACE2 expression.
Dr. Nath, what are the key results and your conclusion?
Thank you, Dr. Garovic. In the healthy kidney, ACE2 expression is abundant, especially in proximal tubules. ACE2 expression decreased in ischemic AKI, was increased in LPS-induced AKI, and remained unchanged in the obstructed kidney. Irrespective of the directional change of ACE2, the MAS receptor protein was induced. Increased expression of ACE2 in response to LPS required heme oxygenase 1.
Can you provide a brief overview of the heme oxygenase system and why is it important in kidney disease?
Heme oxygenase 1, HO1, is an inducible enzyme that protects the kidney against diverse insults. Such cytoprotection occurs as this enzyme degrades heme, an oxidizing metabolite, while generating cytoprotective products such as bile pigments and carbon monoxide. HO1 also promotes cellular iron homeostasis, thereby reducing the risk of iron-mediated oxidative stress in the acutely injured kidney.
Was there anything in particular that made you shift your attention to the heme oxygenase 2 isoenzyme?
HO1 is inducible, whereas heme oxygenase 2, HO2, is constitutive. As we expected altered ACE2 expression, we used as a comparator protein one that's either induced, namely HO1, or one that tends to be unchanged, namely HO2 in the stressed kidney.
What are potential clinical implications? Feel free to speculate on when, how you think it could fit into our current therapeutic armamentarium.
Thank you very much, Dr. Garovic. I think this is a very exciting consideration from these studies. Prior studies indicate that ACE2 may be cytoprotective in injured tissues, including the kidney. This raises the possibility that an AKI not occurring in COVID-19 infection, inducing ACE2 and/or administering its product, angiotensin 1-7, may offer therapeutic strategies. However, in COVID-19, inducing ACE2 is potentially quite dangerous since this would promote cellular infection by the virus. This risk may be circumvented by administering the putative cytoprotectant angiotensin 1-7.
So, what are the next steps?
The next steps include examining the functional significance of ACE2 in AKI by genetic and pharmacologic approaches.
In conclusion, could you provide a brief summary of this work?
Thank you, Dr. Garovic. I will recap these essential findings as follows. ACE2 expression is abundant in the intact kidney, but is decreased in ischemic acute kidney injury. LPS-induced ACE2 expression requires heme oxygenase 1. Mass receptor protein expression is consistently increased in ischemic and LPS-induced AKI, even though these models exhibit divergent changes in ACE2 expression.
Congratulations on these exciting findings, and I'm looking forward to continuing our collaboration in this particular field.
Dr. Garovic, my sincere thanks for your input into this work. It fundamentally improved what we eventually completed. I thank you for what you've done for this study, and importantly for participating in this podcast for Kidney 360. Thank you.
Thank you.
This podcast is copyrighted by the American Society of Nephrology, all rights reserved. All content in this podcast is for informational purposes only and is not intended to be medical advice. This podcast should not be used in a medical emergency or for the diagnosis or treatment of any medical condition. Please consult your doctor or other qualified healthcare provider if you have any questions about any medical condition, or before taking any drug, changing your diet, or commencing or discontinuing any course of treatment. Thank you for listening to this podcast from the American Society of Nephrology.